Warm welcome to everybody joining us today for our 9th annual D3 meeting, where we're going to be covering drug discovery, diagnostics and development. Today obviously is day one. We've got a packed agenda in front of us. I know a lot of you have just come from the Kigali summit, and we kind of wrapped a lot of the kind of output from that and tried to frame it in this kind of discovery, diagnostics and development frame. We've had sessions planned for today, 3 sessions. So we're starting with pipeline access financing. We're looking at the challenges in terms of bringing new discoveries, innovation within NTDs. And then we're going to move to innovations within data collaboration for future NTD treatments. Some very interesting platforms that are kind of out there, and we want to bring some of that into that session. And then we'll end the day with a disease focus, looking at sleeping sickness, the success story within that with the DNDi and Sanofi Pasteur. We're looking at kind of de-risking discovery through partnership and kind of pivot that they've placed in terms of access and the success of that. But so that's the kind of packed day today. I'm not going to go into day 2 with you just now. The program is up, but we're kind of pressed for time. So I'm going to just a quick hello to the panelists for the first session. We're honored to be joined by Professor Kishor Wasan. He's the co-director and co-founder of the Neglected Global Diseases Initiative at the University of British Columbia, and he's joining us very early in the morning from out in Canada. So hi there, Kishor, and thank you for joining us. We've also got Dr. Atteneri López-Arencibia from the University of Laguna in the Canary Islands in Spain, looking at a kind of deep look at their kind of acrotin nitrile derivative leishmaniasis, asbestos, looking at new treatments and just expanding that, looking at the kind of challenges that they're going to be facing, bringing that forward. Dr. Jonathan Marchant, professor and chair at the Medical College of Wisconsin, University of Wisconsin, also joining us from a very early slot, looking at identification of novel modulators of a schistosome transient receptor channel targeted by Carolina Batista, who's part of the DNDi Access Committee, also head of Global Health Affairs at Baraka Impact Finance, and on the international board for the MSF, the Médecins Sans Frontières, looking at mobilizing private capital to catalyze health innovation and in low middle income countries. And then we're also going to be joined at the Q&A by Isaac Chikwanha, the Senior Director for Investment Strategy, Access and Delivery at the Global Health Innovative Technology Fund, the GHIT Fund, based in Japan. But I think Isaac's joining us from the Geneva office. So a very interesting balance within this particular session. I think that's enough from me for now. I'd also like to give a big shout out to from all over the world. And if you could very kindly in the chat box put your name, country and affiliation, i.e. which university or which your affiliation company, or put that in there. It makes for an easier way of kind of recognizing everybody and saying hello. We thank everybody for joining us. I think that's enough from me. And I'd just like to ask the other panelists if you could just mute yourselves and your camera and the microphone mute, and then we'll just pass the floor over to Professor Wasan.
Well, thank you very much. Well, it's a pleasure and honor to be speaking at this prestigious conference, so thank you very much for having me. Kishor Wasan. I'm a professor and distinguished university scholar at the University of British Columbia. I was the dean of the pharmacy school in Saskatchewan, and then upon retirement, I came back to UBC, and I've been running I'm one of the co-founders and a co-director of the Neglected Global Diseases Initiative at UBC. We actually started this organization over 10 years ago, and it's because what we learned, and the byline across our campus was neglected people doing neglected disease research, and they needed a place, a hub, a place to promote their work, to facilitate their work, and so on and so forth. So Dr. Bob Hancock and I, Bob is a world-renowned microbiologist, started NGDI 10 years ago, and at the time, President Stephen Toop, who's actually now in the UK, and and the president at a prestigious university in the UK, he was a visionary in letting us start NGDI. So I only have about 20 minutes, so I'm going to go very quickly through what NGDI is all about. So for us, I think the big thing was, how can the poorest of the poor share in university discoveries? You know, universities are great ivory towers of academic research and development, but the big issue is could we get the great research and that we're doing at the university on the ground, get it translated into actual practice? And so the key was we needed mechanisms for encouraging and funding expensive research and development in neglected diseases. And I think this audience already knows that. And yes, we had just also in Canada had just endorsed the Kigali Declaration, and NGDI has endorsed the Kigali Declaration, which is, I think, very exciting, and I hope will put meaningful resources into neglected diseases. And the other thing was, how do we protect our discoveries early? And I think the most important thing is delivering it on the ground. And so, as I said, 10 years ago, we developed NGDI, and our mission was developing interventions for neglected global diseases and ensuring their delivery to those that need. And you can look at our website and see all the great projects that we're doing. But what we did was we brought together a variety of disciplines, from bench science to biotechnology, pharmaceutical sciences, health, social policy, business, and law together under one umbrella. And our goal was to be really a facilitator of people's research, help them with connections with many of the people that are in the audience today. And, not only didn't we bring our expertise in a number of different disease areas, but also a lot of the research that we were doing, being able to work with partners to translate it onto the ground. Now, this is an old slide from a number of years ago, and I think what the point that this slide makes is that actually neglected global diseases, as the WHO has identified, in 10 years has grown. And that tells us that, even though we're making huge dents, we've got new things coming on. So there's a recent report on product development partnerships that I've given to the other speakers, and I can share for other folks, that talked about some of the partnerships that have been done that have led to the resources to deal with these neglected diseases. And that list is incorporated in that report from the WHO, and that list is over 20 now. Why back 10 years ago it was 13. And so these are some of the funds that we focus on at the University of British Columbia, and of course we also have a strong HIV/AIDS, malaria, and tuberculosis program. Now, why are they neglected? And I think we all know why. And the bottom line is, just to remind the audience, there is a disproportionate level of disease to the resources required to treat the disease. And so over the next couple of slides, what I show you, and I think people know this, that the reason why we use the term neglected is because we just don't have enough resources to deal with these diseases for the magnitude of which the disease has. And therefore, we need to shed light on that. And this slide talks about the importance of the cause of death and how it's had an impact on people's lives. And we all know that heart disease and cardiovascular disease and chronic obstruction disease are major diseases, but we're finding that these neglected diseases and viral diseases and so on are bubbling to the top. And I think if nothing else, the COVID pandemic, and we wouldn't want to benefit over the detriment of others, but I think what the pandemic has shown us the weakness of the world toward neglected diseases. And I think it's given us a renewed interest around the world that we've got many diseases out there that we have to deal with. And if nothing else, it's woken us up. And those diseases have now bubbled up to the surface. So how has UBC responded to this? Talk is cheap. We need to do something about that. So UBC has developed, one was NGDI 10 years ago. One is our University Industry Liaison Office has a global access policy, a global access principle, which is for certain technologies, we will develop it at or below cost for developing world indications. And we're not looking necessarily for commercialization. And many of the people in the audience today, I think, feel the same way and have organizations that align with our global access policy that we developed 10 years ago. And so UBC is trying to do a lot around global access, as well as to engage the next generation of students and trainees to get them involved in neglected diseases at a very early stage. And back in 2013, we were very honored. The University for Allied for Essential Medicine is a group that's been around for a number of years. It was started at Yale University. They have over 100 chapters that are run by amazing students. Did a report on the University Global Health Impact Report Card. And we finished at the top of that list back in 2013, and we got an A-. And you can see some of the grades of some of the other universities at that time. But the reason why we're very proud of that is because it was recognized that our global access principles was taking hold, and people were respecting what we're trying to do at the University of British Columbia. So really the bottom line for us was developing interventions, either therapeutics, diagnostics, telemedicine, social policy. It didn't matter what the innovation was, but the idea was developing innovations for neglected global diseases. But the key issue, the key thing was ensuring their delivery to those in need. It's great to have it in the ivory tower. It's great to have all that research going on. It's great to have proof of principle studies done. But if we can't translate it onto the ground and have it being the new standard of practice to help address these issues, we really haven't been successful. And that's where NGDI has been working on for the past decade. And this is our model of NGDI, which is, interventions with discovery and development and vector control and delivery. And of course, social determinants of health and health equity approach are very, very important. And the bottom line is access, with the result being better health incomes. Now, one of the questions that's come up is, and I think will probably be discussed throughout the next 2 days, is about funding. And 15, 20 years ago, I was pessimistic about this. I was involved in the development of an oral formulation of amphotericin B for leishmaniasis, and I'll show you some of that data at the very end. But 20 years ago, there were not mechanisms to get funding. There were very few mechanisms to get funding. But I have to say, over the last 10 to 15 years, there's been extreme increase in partnerships and funding and success. I think the development of product development partnerships, where we brought together public, private, academic, philanthropic, and governments together to work at a community-based level to be able to move technologies forward and get them on the ground and make an impact, I think has been really, really exciting. I think for this report that I've provided to my esteemed colleagues called Keeping the Promise, that was commissioned in 2021, talks about that over the last 10 years, there's been over 60 new health technologies that have impacted over 2.4 billion people. And some of the great examples are some of the work that was done by the TB Alliance, by Medicines for Malaria, by DNDi and others. And not only did they have health technologies making a difference, but also they had an impact on health systems and global security. So in the last few minutes, I'm going to highlight some of the work, not all of the work, but some of the work that we're doing at the University of British Columbia. My co-founder, Dr. Bob Hancock, is a world leader in addressing antibiotic failure, and particularly sepsis. And here's a very busy slide, and I'm sure we can share the slides with everybody afterwards, where he's been working on sepsis for over 30 years and antimicrobial resistance. But, he's talked about how sepsis cuts over many neglected diseases and things like COVID. And he's done a lot of research in this area and has made significant impact. My colleague, Dr. Richard Lester, who is also the co-director of the NGDI with myself, he's an infectious disease physician, has been working on the ground in Rwanda and many countries for a number of years in telemedicine. And he's developed a program called WellTel Health, where he has been able to make a difference in drug adherence, in diagnosis in the developing world. And in fact, Richard has put together an international program of health research with funders and collaborators all across the globe. And again, it's important that what Rich is doing is that he's translating, the results so that it's making an impact on people when it comes to drug adherence and drug therapy. And that's really quite important, particularly in rural areas in different parts of the world. My colleague, Dr. Catherine Plews, is a world expert in addressing severe malaria, renal dysfunction, and adjunctive therapy, and she's been working for a number of years in different parts of the world dealing with this kind of research. And again, we'll be sharing the slides with everybody so people can do a deeper dive, but it's an example of the kind of work we're doing at the University of British Columbia. My colleague, Dr. David Patrick, is actually one of the world leaders in antimicrobial resistance. And he's been working on preventing antibiotic harm for many, many years, particularly in children and particularly in the area of asthma. And he published an important paper about a year and a half ago in the Lancet Respiratory Medicine in this area. But we're more than just those kinds of things. The former Dean of Economics at the University of British Columbia, Nancy Galini, has been working on the summary of policy instruments for prolonging effectiveness of existing antibiotics. And she is a leading expert in the area of what we call social enterprise. This idea of cost effectiveness, this idea of developing technologies at or below cost for the developing world, where we're not really looking for commercial gain, but we're really trying to make an impact on people's lives. We also have an outstanding center for TB research at the University of British Columbia, and one of the biggest areas, as you know, is drug-resistant tuberculosis. And we've got researchers at UBC that are doing a lot of work in this area and have come up with new algorithms, new therapies, new strategies to deal with drug resistance and tuberculosis research, particularly this area of these XDR and MDRX strains. We have a Center for Microbial Diseases and Immune Research. Vaccine research at University of British Columbia is really quite exciting. And you may or may not know, the Pfizer vaccine for COVID, the lipid nanoparticle, was actually developed by our colleague Dr. Peter Cullis at the University of British Columbia, which was then incorporated in mRNA, which led to the COVID vaccine that Pfizer has been giving out. And of course, Peter has reached international acclaim in collaboration with his colleagues at Penn and in Germany for his vaccine work. But we've had a rich background in microbial diseases and immune research for a number of years. My wife and I are doing research in vaccine development for pertussis and in influenza with Vito Intervac here at the University of Saskatchewan, at the University of British Columbia as well, and Dr. Hancock is involved in that as well. So we've got a lot of exciting work in that area. We're doing work in prevention and control of dengue in Ecuador. Dr. Jerry Spiegel, who is a leading authority in this area, has been doing system work and pilots of integrated community-based approach to reactive insecticide program for dengue in Ecuador for a number of years and has reached funding there. And Dr. Jan Hudak is a clinical infectious disease physician who's done a lot of research in the education side. Course development, educating the next generation of researchers. He's in consulting and so on. So I want to give you a breadth and depth of the fundamental research, the translational research, as well as the educational work that we're doing at the University of British Columbia. And Jan has done some amazing educational programs in this area. As I mentioned earlier, UBC was the first Canadian university to develop what we call the Global Access Principles. And the idea was, through our UAM chapter and Grand Challenges funding, is that for certain technologies that we would develop it on the ground at or below cost for a developing world indication. But when you develop a policy and procedure like this, it's great. But do you have actually success? So in the last couple of minutes here, and I only got a couple of minutes left, I want to give you one example, and that is our work with amphotericin B. So amphotericin B is one of the projects that fit in our global access program. So AMP-B is a drug used for the treatment of bloodborne fungal and parasitic infections. However, the big problem with AMP-B is its dose-dependent nephrotoxicity, that it's given parenterally, it's got low solubility and low permeability, and one of the biggest challenges of AMP-B, and even though the liposomal AMP-B formulations have been fantastic, and recently WHO and DNDi have come out with outstanding guidelines for VL using miltefosine and liposomal AMP-B, but AMP-B does have its limitations in its current formulation. And these, some of the fungal infections like it can be used for, and it's also a very important in leishmaniasis, and I won't go into this in detail by lack of time, but everybody knows the importance of using amphotericin B for the treatment of leash, and the importance of having real-world efficacy on the ground. And these are some of the current treatments for leishmaniasis. And the idea is to be able to develop something that's accessible, that's cost-effective, that's safe, that is tropically stable. And so the Bill and Melinda Gates Foundation provided funding to NGDI researchers in my lab at UBC to develop an oral formulation of amphotericin B that could be cost-effective, that can be safe, that can be accessible, can be tropically stable. And through the Consortium of Parasitic Drug Development at North Carolina, which is where Gates gave the funding, and they funded us. We did animal studies to demonstrate that our oral formulation had almost greater than 99% reduction in the parasitic load in animal models, and at doses that were non-toxic, giving this oral formulation. And here's additional data as well to show that our formulation was very, very effective and, is close to what you would see following an ambisome IV bolus dose. So what we've developed is an oral formulation technology that has now got formulation stability that you can easily scale up. And from there, we move from the preclinical animal studies and the formulation studies into human studies. And of course, the advantages of an oral AMP is it's affordable, easy to store, easy to administer, lack of kidney toxicity, lack of infusion side effect, and lack of liver and GI toxicity. And what has happened now is that we've got orphan drug status from the FDA. We've got patents issued for the technology a number of years ago. But the most important thing is recently we've published now very positive human phase 1A single dose and phase 1B multiple dose safety studies with our oral formulation of amphotericin B. And what we're now looking towards is doing work in systemic fungal infections, in combination of black fungus and COVID, but also in leishmaniasis. And one of the things I've been in discussions with several groups, and I'm hoping to discuss with people today that is there a possibility of getting our oral formulation of amphotericin B out there as a complement to liposomal AMP-B, miltefosine, and some of the other strategies for leishmaniasis in the developing world? But this is a case example, colleagues, of the global access policy. The University of British Columbia, the company that we've licensed this technology to, have all signed on to the global access policy. They don't want to make money from this. They want to be able to develop it at or below cost for the VL indication. So this is a great case example of how the global access policy can work. And we've got several other technologies in the pipeline as well. What we're looking for now is partners and collaborators to take this technology and some of the other great work that we're doing at UBC and translate it on the ground. And my final slide is, we need to have young people. Dr. Jennifer Choi got her PhD a few years ago and is now actually a lawyer. Went back to law school, and now she's a lawyer, and she was one of our leading students in NGDI. And so NGDI has a student group and has their own newsletter. But the key thing is to advocate, educate, and participate. Collaborate outside your discipline to explain your research, and try to get people involved in their own initiation to neglected diseases. I think I'm at the 20-minute mark. I've gone a little fast, but thank you very much for your time, and I look forward to the Q&A.
Thank you very, very much, Kishor, for that wonderful lowdown in terms of the amazing work carried out at UBC, and so very, very encouraging to hear the deep-rooted acknowledgement around access and that kind of foundation that the whole steps that you're taking have been built upon. So that's fantastic, and a lot of resonance in today's world in terms of the access considerations for moving these pipelines forward, for getting that financing, for getting those collaborations. So fantastic overview there, and we really appreciate that. and I'm sure it's going to generate a lot of questions in terms of the Q&A when we come to that. So thank you very, very much for that, Kishor. Much appreciated. And now, before we move to the second speaker, I just wanted to give a quick shout out. We're mentioning access. This is our 9th annual D3. Normally we would have it, face to face as a meeting, after COVID restrictions, we started this online kind of footprint. And for us, it's kind of, in a way, redefined access to medical conferences, because I'll give you an example now. I'm going to give a shout out to some of the people joining us. So Celestine Madugo from the University of Kinshasa in the DRC. Hi there, Celestine. Robert Adunushe from the University of Biu in Cameroon. Dr. Helena Pangaribuan from Indonesia. Back all the way to Brazil, we've got Odalpo Efilua, a PhD parasitologist from the Parana in Brazil. Al Edwards here in Reading at the School of Pharmacy in the UK. We've got Gert Jan Wijant from the Louvain Drug Research Institute in Brussels, joining us from Albania, Valbona Kijona. We have from Senegal, Dr. Mariama Sene, Rebecca Barksby from the Lancet here in London. From Tamil Nadu, Dr. Narayan Asami, all the way from the south of India there, Cape Town. We've got Maria Zinga, Pakistan's National Institute of Health, Dr. Amina Najam. A lot of people joining us, and I just wanted to give a shout out to that because it really makes for an interesting Q&A at the end, so we really appreciate that. Thank you. Great first kind of scene setting by Kishor, which brings us to the second speaker. We want to have a look at some of these compounds at university level. So we have Dr. Atteneri López-Arencibia from the Institute of Tropical Medicine and Public Health at the University of the Canary Islands, at the University of Laguna in Tenerife in the Canary Islands in Spain. I think that's enough for me, and I think over to you, Atteneri, to discuss your acrylonitrile derivatives and their use against Leishmania amazonis. So over to you. A round of applause and thanks, and I'll just back out. Thanks a lot. Over to you.
Well, first of all, I want to thank to the Society for inviting us to share our results here. Well, my name is Atteneri López, and we're here in the Universitario Institute of Tropical Disease, and I'm going to talk about some acrylonitrile derivatives that we have been testing here against Leishmania amazonensis. Well, as Professor Wasan already was mentioned, well, the Leishmaniasis, you already know that is a worldwide distributed infection disease. And one of the data I want to highlight is that the official data doesn't reflect the reality of the problem. For example, in the Mediterranean basin, there are many countries as Spain that hasn't data available, but we already have had many outbreaks, for example, in Madrid, but there are already groups of clinicals working on collect all this data to tell to the World Health Organization to upload to the web. The main problems, as Professor mentioned, are the lack of an effective vaccine, and also that the correct treatments will have this problem with the side effects, the long term, the intravenous, and they are quite expensive, and also the appearance of resistant strains. So what we try to do in our university and in our lab is to check on new compounds, not only synthetic like these ones, but also from plants, from algae. We test many kind of molecules and extracts to check on their activity to test again the Leishmania. So here we tested 32 acrylonitrile against Leishmania amazonensis. Even against promastigotes and mastigotes. And then we conduct the cell studies to check if they were inducing necrosis or apoptosis on the parasites. And I will explain you later why. Well, the acrylonate trials has this kind of a structure with the cyano group, the testosterone, and then we, the chemists, have added different substituents in this radical. And in the bibliography, we already have seen that this kind of molecules presented antibacterial activity, anti-tumoral, even anti-plasmodium activity. So we wanted to check also on leishmania. These were the molecules that the chemists provide us. And as you can see here, the substituents are very different, ketones, esters, sulfones. And also we have the both isomers, the Z and E isomers. Normally in our lab, as I was mentioned, we receive a lot of molecules. So we try to do first a quick screening with just few wells to check on the antiparasitic activity. Then we have the good ones. We move to check on the cytotoxicity against murine, mammalian cells. And when we have a good balance between these two parameters, that is reflected on the selectivity, we continue to study the induced cell death. Just to introduce you a bit how we work, it's like this. We check on different concentrations of the molecules against the parasites, and we use a reactive that changes the color with the metabolism of the parasites. Then we just measure this color, and we could obtain the IC50 values, that is the values we can compare with each other. For the cytotoxicity studies, it's more or less the same, but with the mammalian cells. And then we also perform studies with the amastigotes forms, that for this, first we have to infect cells with the metacyclic leishmania, and then wash, and the treatments, incubate, and then we just release the leishmania by blocking the macrophages, and then we proceed by the same procedure. Well, focusing on the results we obtain, we can see here a huge table, sorry, but it's just to say we have a wide range of activities. For example, we have IC50 from 200 and 1 micromolar. They were two different activities. And then moving to the cytotoxicity, we also have different values, but just to be correlated, if they are so active, they are quite cytotoxic. So in order to compare and differentiate which compounds are the selective one for the parasite, we just determine this selectivity, and we obtain 3 compounds that have a selectivity index similar than miltefoxin, or even higher. So we continue with these 3 for the rest of the assays. These compounds were these 3 you see in the screen, and we also perform the amastigote activity to check that the activity already exists. Here we obtain that the amides were the more active ones. So when we want to continue studying the cell death, why we do these studies always? Because it's too different if we obtain necrosis than if we obtain apoptosis. The necrosis normally is ends in a cellular explosion that causes a strong inflammatory response. What happens if we, for example, if we have a liver or a spleen full of parasites and we explode suddenly with the treatment of these parasites? This inflammation could be worse than the infection per se. So we already know that the features in necrosis are an increase in the size, dramatically lost in the ATP, the loss of the membrane integrity, etc. And on the other hand, we have the apoptosis, that is a controlled and efficient process that works in an enzymatic cascade and prevents all this inflammatory response. Their characteristics are a decrease in the volume of the cell, decrease in the mitochondrial membrane potential, and nuclear condensation, etc. And although apoptosis or the proteins involved in apoptosis in these parasites are not really well described, or not all are described, it is known that the apoptosis occur in this protozoa. So for study this, we perform another experiments to check on all these parameters or cellular events. We will check here. For example, in this image, you can see in blue the cells that has their chromatin condensated in the nucleus. Here we have at 40 and at 100 magnification. And also in red, we have the cells that are already dead after this event. This could be indicatory of apoptosis. In another hand, we have, or here we have the study of the mitochondrial potential. We observe this decrease in all the treatments by a 30 and almost 70% regarding to the controls. But on the other hand, here we have the ATP levels that they appear to be not too really affected. So it could be that the mitochondria, in this case the kinetoplasm, is working, so has energy to perform all this cascade involved in the cell death in the case of apoptosis. Here, for example, we have the analysis of the plasmatic permeability. And in green, we have the cells that have this permeability increase due to the dye, because the dye used is impermeable to the cells, but when the permeability is altered, it gets size, it binds to the DNA, increasing its fluorescence. So here you can see how in the 100 magnification, the nucleus and the kinetoplasm in green fluorescence. And also, if you check it, also the shape, the integrity of the cell, it seems to be intact. So this could be also, this could tell us which kind of cell death is happening on the cells. And here to finish, I have the results for the reactive oxygen species. As you can see in the 40 magnification, we have the Q31 appears almost the same than the positive control, with almost all the cells accumulating this species. Normally, these species are produced or are accumulated because of stress. This stress, in this case, is supposed to be caused by the treatment. And this stress also induces the apoptotic event via mitochondria. That is one of the intrinsic via of to activate all the enzymatic process. So just to mention, in a previous work, we also tested all these acrylonitrile against another parasite, Trypanosoma cruzi, the one that causes the Chagas disease. And we obtained also that 5 of them induces this kind of cell death on Trypanosoma cruzi. But just in common with Leishmania amazonensis, we have the number 29. So just to conclude, these compounds show activity against Leishmania amazonensis as well as Trypanosoma cruzi, and observing that the amides and the phosphosubstituted ones were better than the rest of the substitutions. Also, if we observe the stereoisomerism, also the isomer is used to have better activities than the Z isomer. And we observe good selectivity indexes in these 3 molecules that were even better than miltefosine. Then with the cell death studies, we can conclude that at least these 3 compounds induce this kind of cell death that could avoid this inflammation. But also we have to continue the study because there are no relation on the behavior between these 2 genera of kinetoplastics. So maybe we can continue working and checking on this. This is what we have done with these 2 works, and I want to thank all the lab team that works really hard, testing on many molecules, extracts from different sources, and also want to thank the chemists that provide us with all these compounds. And I also wanted to mention that we also work not only Leishmania amazonensis, also Leishmania donovani, also we test molecules against different free-living amoeba that causes important diseases like encephalitis. And there is still a lot of work to do, as Professor Wasan was telling, to fight with these diseases. Thank you very much. If you are, I'm open to the questions at the end of the session.
Thank you very much for that presentation. I hope you can hear me. Yep, you can hear me. Excellent. Brilliant. Thank you very much for that itinerary. I'm sure the pathways and the kind of that your workflow has a lot of resonance with a lot of the audience in terms of developing at university level a series of molecules, and the questions I think really going to come through in terms of what are the challenges that you're going to face and how do you move from that to forward. So I think that's a very interesting, that you've got activity there, all the tick boxes are being ticked. How do you progress that? I think is going to be part of the Q&A. So we really thank you for that deep look into your acrylonitrile derivatives and the excellent work that you're doing there. So thank you for that. That takes us on to our next speaker, Dr. Jonathan Marchant, actually Professor Marchant, one of the chairs there at the Medical College of the University of Wisconsin. And I'm just going to, because of time, just quickly pass the floor over to you, Jonathan, in terms of your talk on identification of novel modulators of a schistosome transient receptor potential channel targeted by praziquantel. Over to you, and a round of applause.
Thank you, Cameron, and thank you everyone for coming today to this panel. As Cameron said, I'm a professor at the Medical College of Wisconsin. I'm a basic scientist. I'm very much at the discovery end of the spectrum here, involved in early stage drug development, and it's great to be at a conference and a panel like this to help us with our work and make new collaborations. So what I'm going to talk about in brief presentation today is our work with this drug, praziquantel, which is very widely used for treating schistosomiasis and other diseases caused by parasitic flatworms. Praziquantel is a very old drug. It's been around since the 1970s. And it's surprising, as it's one of those FDA-approved medications that we actually don't know how it works. And this has been a roadblock for rational drug design and development for quite a while, and a problem in the field to try and figure out how this drug is working, so we can, on the one hand, better do surveillance for drug resistance if we could find a target, but also design better agents that either interface with the target or target the same pathway that parasites are vulnerable to. So praziquantel is a very simple drug. It's most commonly known as treatment for schistosomiasis, but it's also effective against many other trematode flukes, as well as being effective against tapeworms. One little quirk, which I'm going to come back to a little later in the talk, is that praziquantel is well known to lack activity against one particular type of liver fluke, fasciola. So praziquantel doesn't kill fasciola in vitro, and it can't cure fasciola in humans or animals. So what we have known, although we haven't known how praziquantel works for a very long time, we have known right from its introduction key things that praziquantel does. So one of the first things praziquantel does, and we've known this since the 80s, is that it causes a rapid contraction of parasitic flatworms. So these are contracted schistosomes on the left, and on the right, this is some old data showing a strong, sustained muscle contraction in flatworm muscle. When you add praziquantel, the worms contract and stay contracted while praziquantel is exposed. The next thing we know is that praziquantel also causes surface damage to these worms, and that's very important because the surface damage likely exposes worm antigens to the immune system that then is part of the cascade of effects that leads to worm elimination, schistosome elimination from the body, this muscle contraction and then a sustained surface damage. And the final thing we know is that these effects that you see, these phenotypes, are quite stereoselective. So contraction and surface damage is caused by the R enantiomer of the drug in the racemic clinical formation. Yes, enantiomer also causes this effect, but at much higher concentrations. So the sort of third thing we've known for a very long time is the stereoselectivity of these effects is predominantly caused by R praziquantel over S praziquantel. So our contribution as a lab a couple of years ago is to find a target in parasitic flatworms that mimicked many of these characteristics. So this was our paper that we published a couple of years ago, that we found that praziquantel activates a schistosome ion channel. It's a transient receptor potential channel, a TRP channel. These channels are very often involved in sensory physiology. And if you see at the bottom, this channel was stereoselectively activated by R praziquantel over S praziquantel. And the piece of data on the bottom right shows a sustained calcium influx caused when praziquantel activates that channel. So that sort of mimics the effects of praziquantel on the worm in terms of muscle contraction. So we were quite excited about this. This sort of finally gives us a candidate target for the drug in the worm. And since we published that paper, there's been a variety of other studies that sort of add more fuel to the fire, that this could be the long sought after target of praziquantel in parasitic flatworms. So in collaboration with Merck KGaA in Darmstadt in Germany, they gave us, as only Merck could, their sort of back catalogue of praziquantel analogues, and we could do SAR on the channel. And the pharmacology of this channel in vitro mimics almost exactly that of worm contraction, suggesting that is truly the target. Then, really quite bizarrely, we found that in fasciola, the ortholog of this channel is not activated by praziquantel at all. So that was very consistent with praziquantel lacking effect against fasciola liver fluke. And that while fasciola is not activated by praziquantel, is it has a single amino acid change in the praziquantel binding pocket of this channel. And remarkably, if you change that variant residue back to the residue that's present in the schistosome channel, then it becomes activatable by praziquantel. So that's it, yeah, sort of a remarkable piece of data that the fasciola channel doesn't work, but we can make it praziquantel sensitive. And then work by Tim Anderson's lab in San Antonio showed that in a strain of schistosomes engineered in the lab to be less sensitive to praziquantel, that this channel was the top genetic association in a GWAS study. And then finally, we did a very small drug screen on this channel, and one of our top hits that came back was also a compound that had been found in phenotypic screening of schistosome worms. So the fact that you get the same compound out of phenotypic screening, just looking for compounds that kill worms, and then we can show that that compound is a different chemotype that activates the channel, is also, I think, quite good evidence that this is a very druggable target and likely the relevant in vivo target of praziquantel. Okay, so we think that this particular ion channel is a good target for looking at, to coming up with new chemotypes to tackle diseases caused by parasitic flatworms. So what our lab is trying to do is, now we have a target, we know how praziquantel binds to this channel. I've shown you on the left that praziquantel works on schistosomes. On the right, it doesn't work on fasciola because fasciola is missing a key hydrogen bond in the praziquantel binding pocket, and that difference is sort of taunting us to try and derivatize praziquantel to find analogs that can remake that interaction. But also we can map binding pockets in cestodes and monogeneans, which also cause different human and agricultural diseases. Those binding pockets are also slightly different. So that's an idea that we may be able to identify drugs that work back better than praziquantel to target particular parasites. Option 2, which is maybe a bit more of a holy grail, would be to identify through target-based screening some broad spectrum drugs that, irrespective of binding pocket variation, work on all different sorts of parasites. So we have a chemist in the lab who's sort of working on both these projects, trying to dink around with praziquantel to come up with better analogs for different parasites. And then secondly, we've just finished a unbiased high throughput screen to look for completely new chemotypes that engage this ion channel in different sorts of parasites. And this has been a fun assay to put together because the channel actually has a very big signal to noise. It's easy to work with. We've been able to miniaturize this assay down to 6 microliter volume, and we've just finished a 600,000 compound screen. This is a little bit of a challenge for a small lab because we have to put together all the sort of collaborations and validation assays, but we're sort of logically doing that. We're screening them against this ion channel in our parasites, trying to build collaborations to test chemotypes on different sorts of parasites before moving to in vivo. But the hope is we may be able to come up with new chemotypes that have anti-parasitic activity. And I'm just going to finish. I'm not going to go through all the data from a screen in a short talk, but what this graph sort of summarizes the results of the screen. On the left-hand axis, we have sort of potency of activators and inhibitors, and on the right and on the bottom axis, we have sort of top compounds in the screen. And for agonists, we are able to find things that are more potent than praziquantel in activating this ion channel. And perhaps more remarkably, for antagonists, we're finding compounds that go down to nanomolar potency for channel inhibition. And if you look at, for instance, data from praziquantel from that screen, you can see in the blue curve, it's activating the schistosome channel very well. But the light blue, it's not activating fasciola at all. And if you look at if we sort of look at potency and look at a few different chemotypes, here's one chemotype that is able to pick up both schistosome channel and activate the fasciola channel. So that would be exciting for us to try to get onto fasciola to see if that's a completely new treatment for that liver fluke. And here's an example of another chemotype, which for schistosomes is more potent than praziquantel, while still maintaining good activity at fasciola. We're just a schisto lab, but we can throw all these things on worms, and just like praziquantel that causes contraction and surface damage, both these chemotypes, at least on schisto, do exactly the same thing. So from target-based screening, we can sort of come up with compounds that thankfully seem to preserve activity. So I'd like to finish We have thankfully provided the field with a good candidate channel, this TRPM praziquantel, which I think is a very validated target for treating parasitic flatworm disease. We've got a lot of new chemotypes that have come from that screen that hopefully show activity against this ion channel orthologs in many different species and hopefully against parasites themselves. And as I said at the start, we're very much a discovery lab. So this is great while conferences like this and panels like this, because it's very easy for us. It's a good problem to have, but we've sort of swamped and overwhelmed with chemotypes and choice and how to progress these basic science discoveries and evaluate them towards SAR and clinical progression. So I'm going to end there, and I would be happy to participate to the discussion at the end of the session. Thanks.
Thank you very, very much, Jonathan, for that wonderful presentation. And you get a real sense of excitement in terms of where you are in this new target, the TRPM target. Interesting that you've aligned, or you've got in Merck are giving you the various derivatives, and I wonder how that might progress in terms of co-developing this. I'm sure that'll pop up in the Q&A at the end, but thank you very, very much. That's fantastic. Thank you very much for joining us at such an early hour as well. So thanks for that. That brings us very nicely and neatly. We've heard a little touch in terms of two very exciting pipelines there from Atteneri and from Jonathan, and the overall kind of desire for collaborations and partnerships rooted in access by Professor Kishor Wasan. That brings us very nicely, I think, into our next speaker, which I'd like to introduce, Dr. Carolina Batista. Carolina doesn't need much introduction, because obviously if you're in global health, you know how much Carolina has been involved in this sector. I had the good fortune of meeting Carolina way back in 2017 when we put together a Chagas disease meeting in the States. This is prior to the priority review voucher being awarded to the DNDi for their Chagas disease compound. Carolina is also on the access committee for the DNDi, and also the international board member for the MSF, as actually here in terms of her involvement as the head of global health affairs at Baraka Impact Finance. We've heard a lot about pipelines. How do you actually mobilize capital? And Carolina is going to speak about mobilizing private capital to catalyze health innovation in low middle income countries in terms of getting to the NTD goals. Again, enough from me. I'm wary of the time, so I'm going to just give you a round of applause and hand over the floor to Carolina. Carolina, great to see you, and thanks for joining us.
Thank you, Cameron, and thanks everyone for the great presentations before. It's great to be in the discussion on NTDs, which is a big passion of mine, and thank you for the Society for the invitation. So as Cameron said, I'll be speaking today about the role of private capital to catalyze health innovation in low middle income countries, in NTD endemic countries, but also as a path to achieve the very lofty but achievable NTD goals as they are in the NTD roadmap. So what makes I'm here today, as Cameron said, to talk about the private capital, but specifically about the role and the work that Baraka Impact Finance does on this field. So what motivates us at Baraka is really the achievement of the Sustainable Development Goal 3, which is health and well-being for all, which includes, as we all know here in the panel today, ending the epidemic of AIDS, TB, malaria, but also NTDs, and reducing morbidity and mortality. Unfortunately, the bad news is that the funding gap for the Sustainable Development Goals 3 is increasing, and it's expected by 2030 to have a funding gap of about 370 billion annually to achieve that. And this is estimates before COVID, which now we know because of all of the consequences, the social, economic, and health consequences of COVID, these estimates might be even larger than that. In an era of declining official development assistance, donor money, this gap will need to be filled with greater, not only domestic resource mobilization, but also with greater engagement and investments from private capital markets, which nowadays are estimated to be about $200 trillion. So just a small portion of that will really successfully address all of these funding gaps. And we've seen from the previous presentations how amazing work is being done at the university labs by professors, by researchers, and this will require a lot of funding to address the goals. And we really believe that private capital holds the key to unlock all of this potential. So needless to say, the COVID-19 pandemic has really had an unprecedented impact in not only in health, but in social, economic, but mostly for the world's poorest. We've seen how the world's poorest have been disproportionately affected by the pandemic, but also I think that the pandemic has really underscored the health system's underperformance and really, as many are calling it, the 4 I's of ineffectiveness, inefficiencies, inequities, but also inadequate responses. And this is primarily due to the long and chronic underinvestments of health systems, but also not only the underinvestments, but suboptimal investments on health systems, especially on weak primary healthcare services in endemic countries for NTDs that really have left a lot of people out and really has generated a lot of a big impact in the world's population, but also really risking reversing all of the progress that we've achieved in the last years in controlling and elimination of NTDs, primarily due to generous donor funding, corporate responsibility initiatives. But now with the straining of the health systems and donor money, we'll need to mobilize more capital, and the funding gap to fund those elimination and control goals will get bigger and bigger. So what do we do? And so it's critical to secure the investments to achieve the roadmap goals and move towards sustainable development goals, not only on health, but as we know, if we invest in NTDs, it will inevitably improve water and sanitation, gender, education, and all of that. So NTDs is really at the core of the Sustainable Development Goals as a marker for development of the population, but especially for the world's most vulnerable. So when we look into the new NTD roadmap, there have been strategic shifts, and really I think these strategic shifts, as I was reviewing them recently, they really provide a frame to guide investments and financing for NTDs, and it really has to do with the impact measurements, and when you think about now of innovative financing, of impact finance, how does that impact? How does the impact of the NTD programs will be not only quantitative but also qualitative, looking specifically into who's accessing the programs from an equity perspective. Equity now is really high in the global health agenda because of COVID. We've seen how equity has, or the lack of, has really played a role in leaving a lot of people out of innovation, of vaccination coverage, etc. But also when you look into one of the other elements and the shifts from that are the core of the NTD roadmap now in terms of cross-cutting approaches. So how do we use investments and mobilizing more investments, both domestic but also private investments? How do we use that as a means or as a foundation for building more resilient and stronger health systems? We've seen how the weak health systems had really jeopardized the world's response to COVID. But also, when you think of one of the core elements of the new NTD roadmap is from program and donor ownership and priority setting to country ownership. And I think this really sits very close to my heart. And I think that really provides us a strong frame into how can we mobilize country and domestic investments to fund all of these initiatives that we've seen before, but also how can we maybe think of of donors and investors from endemic NTD endemic countries to fund those innovative tools that will not only help people to be healthier, but also break the cycle of poverty that is really trapping a lot of the people in those countries in this poverty trap. So what does Baraka do, and how do we think that mobilizing private capital can really help finance all of the not only the SDGs, the Sustainable Development Goals, but also the NTDs targets? Well, we really bring a team with cross-cutting or cross-sector expertise from global health, like myself. I'm a physician, and I've been working for many years in NTDs and access to care, but also we bring people with the strong impact finance backgrounds to help us understand where the overlap of these two themes that for many people they're not together, but they're really very strongly linked, and especially now when we see that the funding gap really means that people are very far from reaching the health innovations they need. We also bring over 250 pipeline companies and a portfolio of companies that are most of them based in endemic countries, in NTD endemic countries, such as Latin America, where I come from. I'm Brazilian, Asia, Sub-Saharan Africa. And these companies are run by people, entrepreneurs, and brilliant people who really are dealing with the day-to-day challenges of diseases like malaria, Chagas, leishmaniasis, and a lot of these, and the challenge of delivering healthcare. So they know these innovators, they know what they're dealing with, and they're really building context-specific solutions. We also believe in the power of data, and we've seen now how creating a strong data-driven dashboard can help investors who are oftentimes not familiar with global health, even though now anyone opens the newspaper and sees health, global health, investments, and WHO. But for a lot of investors, global health is still a foreign concept. So really creating a data dashboard that translates health indicators, global health indicators, but also macro data such as capital concentration, access to care, access to capital. How can that kind of analysis and data help investors de-risk their investment decisions, but also a guide? If, for example, an investor wants to focus on gender-specific health innovation, so how does that translate, for example, into NTDs and what kind of NTDs have a gender focus, etc. So, and we have engaged over 700 impact identified investors, and that means from private investors, from family offices to large foundations and others. So that combination really brings a strong ecosystem that can really catalyze those health innovations reach those who need in countries. And this is just a snapshot of the kind of companies we're talking about. And these are, as I said, companies run by entrepreneurs that are really tapping into the information, communication, and technology revolution that's happening in a lot of these endemic countries. We've seen how COVID has really catalyzed and really accelerated the development, but also implementation of, for example, telemedicine, applications, phone applications, mobile technology, and really bringing health solutions closer to communities, not meaning that people will have to go to hospitals. A lot of the hospitals or healthcare centers during COVID have closed or have decreased their activities. So people had to innovate, and problems, and sometimes a big challenge brings opportunities to innovate. And we've seen that with COVID. And there are a lot of solutions that are really using the power of mobile technology. There are billions of people with mobile phones in low middle income countries, in NTD endemic countries, that we've seen also solutions that have been done, for example, using artificial intelligence to help diagnose leprosy, for example, between Fiocruz, Novartis Foundation, Microsoft. So there are a lot of future-driven solutions to tackle those types of diseases. A lot of clinical support tools are being developed by the same entrepreneurs in those countries. A lot of data is being generated. but this has to receive funding to be able to scale and achieve more people. So we need also to understand that all of the solutions are being not only developed and implemented in NTD endemic countries, in low-middle income countries, but they're also a strong way to collaborate into creating more resilient, but also stronger health systems, and especially primary health care. A lot of these solutions are really bridging the gap between people who until now did not have access to healthcare, and now because of these innovative solutions, they are achieving better health, more preventive measures because of those innovative solutions. But to do that, we really need to mobilize capital to do that, and also to help these solutions pave the way and make the NTD goals achievable by using those types of technologies. So I think the takeaways here, and that's my last slide, but it's really controlling and eliminating NTDs is core to achieve the Sustainable Development Goals. As we all know, the global health funding gap is really huge, and it's increasing, and it's expected to increase. COVID-19 has really impacted the NTD programs and all of the progress that has been achieved in the last years. And private capital represents unique opportunity to bridge those global health funding gaps, but also to achieve the NTD targets. Health innovations is happening, and it's happening at fast speed in low-middle income countries, fueled by this technology and digital health revolution. Entrepreneurs are really doing the best they can, and really putting all of the resources together to help people have better access to healthcare, but not only better access, but in affordable and more quality-driven ways. And I think that for me, the future and this type of future tech-driven digital solutions really have the capacity to delete all of these Asian diseases like NTDs, leprosy, schisto. We've heard leishmaniasis, people who are disfigured in these countries who can really benefit from all of these innovative tools. So mobilizing private capital and mobilizing domestic funding will be a key to achieve that. And I'm happy to answer to your questions in the Q&A. And thank you again for the opportunity to speak today.
Thank you very, very much for that, Carolina. Great presentation, and a fundamental point there. And obviously, you mentioned the releasing of and the whole thing about private capital, sovereign capital in the endemic countries, or in this kind of move by the WHO to promote country ownership. I guess hand in glove with that comes the political will as well, which is something else that we probably weave into the Q&A. But fantastic to see the Baraka kind of approach to all of this. There's some questions already coming through. I think on that note, if I could ask Isaac, I don't think you've got a presentation, but I'm going to introduce you in any way. Is that correct? Yeah. And then I'll just, if all the panelists could come back, I'll introduce Isaac, and then we'll get stuck into some Q&A. And I've been told we can run it until 1:25, maybe just 1:25, 1:28 before the next session starts. So a little bit over time. But firstly, thank you very, very much for all the great presentations, and to the audience for their questions that are coming through. I'd like to introduce Isaac Chikwana. He's the, as I mentioned earlier, from the Global Health and Innovation Technology from the GHIT Fund. He's actually in Tokyo, not Geneva, which I didn't realize. Sorry about that, Isaac. So you've got a late start. Everybody's got an early start. Isaac, the Senior Director for Investment Strategy and Access Delivery at the GHIT Fund. So a warm welcome to you, Isaac, and thanks for joining us today. If it's okay with the panelists, I'm just going to kick off. We've had some questions come through, and I was going to weave it into a kind of narrative, if you like. In the original slides, Kishor, you mentioned in your slide pack very powerful words: advocate, educate, participate, collaborate. I love that. Just picking on the educate part of that out, we've had some questions come through, and just to come to those questions. So we had some questions come through around on the education side of things in terms of mentoring. In terms of mentoring, I know you've answered that, but I just wanted to kind of widen that to everybody else and spin that on its head somewhat. The question itself came through from Ifeola Oladapo, who was asking, is it possible for research scientists in different countries to have this mentorship program as a young scientist? Is asking that question. You've answered him, absolutely, yes, we can, and it's something you'd already mentioned. I want to spin that question on its head. We're talking about mentorship from university to researcher. Do we need mentorship from the funders? So GHIT as an example, and let's say Baraka in terms of the platform that you have. Do the university researchers need mentorship from people like yourselves in terms of how do you streamline your research to get to the funding?
Actually, and it leads to, Carolina's excellent presentation. I think the number one frustration for universities and for researchers at the university is it's great to know that there's all these potential places to get funding and resources and partnerships, but how to start? Who do you talk to? How do you build that relationship? So do I send you an email and we start talking? I mean, I think that's really the biggest frustration. One of the things that we do at the University of British Columbia is we have a mentorship program where we have young scientists and either at the assistant professor level or even graduate students and postdocs, and we partner them up with more senior scientists to get them engaged in the kind of work that we're doing. So we can certainly expand that to people outside UBC, and in fact, many of our senior scientists do work on the ground in the developing world. Things have changed. We're not in that ivory tower anymore where we're sitting at the big university and that north-south issue that we've always had. Our people are actually working in those countries, and so they can be mentors on the ground in those countries. To spin your question on ahead there, Karen, to answer that, that's how we connect people. But yes, we need to get better. Universities need to be better educated about who to talk to, how to connect with those potential funders, who can help us facilitate those discussions. And so, listening to that outstanding talk by Caroline at the end, my question in the Q&A was, how can we work with you so we can get resources to the university professors? But it's not about just doing the research and the university professors. It's about how can those resources then you take the great work that we're doing at all the universities and translate it so it has an impact on the ground at the places where we need it. So it's that combination of that. We don't want just research to be doing research in our ivory tower. We want to go that next step and take that work and connect it to the ground. So we need to have.
I'm just going to jump. Yeah, and thank you very much for a great answer there from both perspectives. I'll just ask that to Isaac, just to bring Isaac into the conversation a bit. How do you feel about GHIT? I know you're involved in so many different investments and so many different kind of projects. In terms of mentoring universities to get their kind of pipelines out, what are your views on that?
So thank you very much, and my apologies that I don't have a presentation. But let me just quickly start by telling the participants what we do. So GHIT is a public-private partnership based in Tokyo, made up of the government of Japan, Bill and Melinda Gates Foundation, Morgan Trusts, Japanese pharma. And we fund research and development of health technologies like vaccines, therapeutics, and diagnostics for NTDs, malaria, and tuberculosis, and across multiple research platforms, from discovery all the way to clinical trials. And because one of the conditions for GHIT funding is you need to have a collaboration between an international organization and Japanese organization. And most of the studies, like the clinical studies that we do, are conducted on the ground, in low-income countries. And for us, we feel that our funding provides that, maybe it's not mentorship, but we hope that there's some kind of transfer of knowledge to the local researchers and local staff who are involved in the clinical trials on the ground. And some of the organizations that we fund actually work with research institutions in Africa, for example, like KEMRI, IPD in Senegal. So there is that level of transfer of knowledge.
That's a fantastic answer. Thank you for that, Carolina. I know there's a lot of overlap. Well, a lot of positivity from Kishor into the presentation and the kind of Baraka approach. I just want to bring in the other two speakers as well, because there was a question, an interesting question from Dr. Janice Lasdens in terms of the acrylonitrile compounds. And the question was, how do you foresee this moving forward in R&D? Do you think these will be taken up? But I'd like to ask that question both to yourself, Atteneri, and to Jonathan, in terms of how do you both see your work progressing? Jonathan, you've got the kind of thing with Merck there, a kind of little bit of an inroad there in terms of the derivatives. But I'll ask that question first to Atteneri. How do you see your acrylonitrile derivative program or library kind of moving forward, progressing, as it were? You have to your volume. Sorry, yeah, sorry.
Sorry. The only thing we can do more here in the university, or at least with our fundings, is just to perform like animals experiments. I mean, in vivo experiments. We have done some with acrylonitriles and with other compounds also from alga that were so good. But then also, I see it like not a problem, but kind of problem for a challenge. Is that the universities want to protect this resource as a patent. I mean, so at the end, we cannot share too much information to the community, and I see it like a block between the community. I mean, this kind of, I don't know how to say this, procedure in the universities these days.
But I think the only thing is
the
IP position. Yeah.
The only thing is just to protect, and then to wait or look for a company that want to buy this protection to develop the treatment or the formulation.
Okay. So that's reaching out to the right
people. I mean, I think this is a great discussion. I think there's a long, deep valley between discovery research and progressing things towards the clinic. But I think in academia, we have to do as best we can in terms of validation before we pass the baton to others who have better expertise to take things forward. And I think panels and conferences like this are great for making new connections and helping catalyze that sort of research and interest. I mean, I think we've heard a lot about private capital, and big and small pharma also may have interest depending on their priorities. So I think we have to do what our skill sets are best, which is continue doing research and also convey our knowledge to the next generations.
Fantastic answer. Thank you for that, Jonathan. Just coming back to Carolina there for a second. Another question came through. We've heard here really about how companies might get involved, and obviously private capital has to get involved. The question was, what is the return that your investors are expecting? We're all here because we care. All of us are here. There's compassion. We want to see a better world. How do you frame that to release private capital to get into universities, Carolina?
Yeah, no, I think that's a great question, and I think it's been in a way my personal mission on that, because having worked for so many years with NTDs and being on the ground, and now talking to people who not always understand exactly the challenges of NTDs and the patient journey in, I don't know, in Kinshasa or in a rural area of Latin America, or, you know, so it's really trying to translate that. I think the self-identified, let's say, impact investor has different expectations in terms of return. If you compare, for example, with a typical venture capital investor, right? They're really looking into 10x, 100x, etc. I think the impact investors, they have, not that they don't have expectations, they have expectations, but they really look very much into the impact metrics. And I think one of the core features of Barak, and what we really strive to do, is really to translate health indicators and all of those data into impact metrics, you know, and really trying to see to help investors see that, for example, the reduction in morbidity and mortality can be translated into economic gains, but development gains, and how that will pave the way, for example, for you know, better development, but also for more economic and thriving societies, right? So, and I think, again, and I think that's why my presentation, I really focus on the role of really investing in those innovators that are on endemic countries, because these people are just not conforming to the status quo. They don't want to accept that, you know, healthcare in their countries has to be suboptimal, has to be of low quality, unaffordable. So they're doing that. And I think just to get back to Professor Wasan's question, I think, you know, we talk a lot to universities and people who are developing tools and health tools, not only on drugs. Drugs is still, I think it's our next frontier, and I hope we get there, because that's something that's very close to me after so many years working with the NDI. I think, you know, we have a lot of people who are developing at university level, let's say, tools, or you know, the other day we spoke to somebody who did like a obstetric tool out of a university in the States that was really great for, you know, reducing mortality for women. So, but I think drugs will be the next phase, and it's just the fact that, you know, it requires more funding and requires more investment. And I think we have to maybe change the narrative and create some literacy. We talk a lot about capacity building for endemic countries, but I think we have to do a lot of capacity building for the investors also. You know, why investing in drugs is so much needed, and especially for NTDs, because it's the result of market failure. So we have to change how we talk about it. But I think it's great questions, and I'm happy. I'll put my email on the chat, and happy to continue this conversation also on different channels. And thank you, Cameron, for facilitating the discussion. Oh, you're mute.
You're mute.
Cameron, you're mute.
Sorry, there we go. I'm so sorry. I was supposed to not do that. We're coming to the end. We're basically out of time, which is always the case. But I just wanted to ask a last kind of question, or actually wanted to put this to you, Isaac. As a funder, what would be your advice to Jonathan, to Kishor, and also to Atteneri in terms of, you know, I don't know, pushing their pipelines or their work forward? Would there be any kind of advice, in a nutshell? Because we've got to kind of.
Yeah, I mean, it's probably something that they already know and they're already doing. As was mentioned already, it takes a long time to take an innovation through the R&D process and all the way to manufacturing. But I think one of the reasons why the PDB model has worked is that collaborative research approach, where the academic institutions are engaged with potential manufacturing partners and other funders and stakeholders into a partnership towards developing a product. So I think taking that approach could really accelerate things and help along the access value chain. But I think they're already doing this. So it's not really hard.
Can I add one thing? I know you're over time, but I'll be brief. I think it would be valuable if we could get resources of who we should contact and list. So we know who to talk to, because I think the biggest frustration for many at the university level is who are the key people to talk to help facilitate those interactions. I mean, researchers, Jonathan's a researcher, I'm a basic scientist myself. We're all researchers, and our goal is to do the work and, have others find us the connections. I mean, one of the reasons why we started NGDI was I watched the frustration for 20 years of all of us finding who are the right people to talk to about getting our work funded, and more importantly, getting it translated. And that's why we started NGDI, to find those avenues. So it would be great if we could get a list of who to talk to, and then have those initial conversations, not just emailing and then having nobody contacting us. I think that's frustration.
That's a great answer, a real world answer, and I think echoing in terms of the how other people are trying to kind of push their pipelines forward.
I've been doing NTDs for over 30-some years, and I've heard the same thing over and over and over again. And, I don't want to be a crotchety old man. I'm very positive and optimistic, and I love what I've heard today. But, I mean, it's time for action. Yeah. I mean, everyone talks and talks and talks, and we just can't get people connected, and we need to get the right connections, you know, so that, that fit goals on both ends. Because sometimes as university people, we are also not in tune to what's really happening on the ground. So I like the comments that both our funder colleagues have made. We have to be, relevant, and that's important.
And potentially bringing in access to the way that you're kind of designing your work or thinking about your work. Maybe that's going to be a kind of a catalyst for these funders to come on board. If you look at the PRVs that have been awarded the priority review vouchers in this space, even though the priority review voucher does not have an access consideration, it's the access programs that have actually won it for, in various kind of disease settings. So that's food for thought. I'd like to say thank you very, very much. Unfortunately, sadly, we are out of time, and we're going to have to come to the next session, which the link is up. We're a little bit over, so I'm going to get a lot of trouble probably after this. But thank you very, very much to all of the panelists, Isaac as well, for joining us and giving your input there. We need longer, right? We need longer, but you heard the lack. We need people to come together, and I really do hope, especially from the wonderful kind of participation and questions coming through from the audience, that a lot of forward movement comes as a result of this session. I know you're all contactable, and if we get any, we'll push forward to you as well. People are already saying thank you very, very much for a brilliant session, and so genuinely thank you very, very much, and thanks for answering the questions as well. If you can join us for the next session, it's going to be looking at data collaboration platforms and the future. So that's the next session. I'm going to jump to that now. I'm going to have to say adios. Thank you very, very much for the early starts and the late starts. Very much appreciated, and I do hope this translates in a forward movement for everybody present. I'm sure it will. So thanks a lot, and over to
thanks everyone.
Thank you.
See you in the next session. Thanks a lot. Thanks a lot. Bye-bye.